Hi everyone. So research has shown that students perform better if they study in an environment similar to the room where they take their exam. If you recall our first exam, the room is very quiet. So you can learn to study in a room that is quiet to tend to do better on the exam and feel less nervous when you're taking it as well. Now, chapter 13, we're finishing up talking about heart and circulation, cardiac physiology. So some fun facts is that the average heart rate's around 70 beats per minute. So by the time you're 70 years of age, your heart has beat over 2 billion times. Now, there's quite a few good lectures and videos on YouTube that talk about heart and heart physiology. So I've linked some here, including one on the EKG or ECG that we'll talk a little bit more about in lab. But these are some nice links to go through to get some basics of heart physiology down. And so I'll remind you of your anatomy at this point, where we're talking about the heart and the associated blood vessels, right? And remember that in humans, we've got a four-chambered heart. And so we have two circuits then of blood flow. So we have a pulmonary circuit that goes to the lungs and back to the heart. And then we have a systemic circuit that goes to the body and brain and back, okay? And so if you remember, your right atrium is what's receiving deoxygenated blood from the superior vena cava that comes from your brain and the inferior vena cava that comes from your body. Those empty into the right atrium. That blood then goes to the right ventricle. It's pumped out the pulmonary circuit then to the lungs where oxygen is obtained, carbon dioxide is lost, and then that oxygenated blood is going to come back in to the left atrium, go to the left ventricle, and then be pumped out the aorta. So you see this ascending aorta here and a descending aorta as well. Another important thing to remember about the circulatory system is that you have these major arteries and veins, and then as they get smaller, you go to arterioles and venules, and finally capillaries. And the capillaries where you have um, a mixture of deoxygenated and oxygenated blood, or the blood is losing oxygen as it travels there um, and is in the tissues, right? So tissues are going to be beds of capillaries that are able to feed the tissues with oxygen and dispose of the carbon dioxide waste. I'll remind you too that in the heart itself, between the atria and the ventricle, there's valves that prevent backflow in the heart. Okay, and so it's a very carefully orchestrated pumping mechanism that keeps blood flowing in one direction and creates the blood pressure we need in order to properly oxygenate and bring nutrients to our tissues. Okay. So when you look at the readings of the heart, we're looking at essentially contraction and relaxation. Okay. So it's called the isovolumetric contraction when the ventricles contract. And at this point, this is the right ventricle. At this point, this atria is relaxed. So blood's going to come into the right atria, go into the right ventricle will pump. That's an isovolumetric contraction. Okay. Then ejection, as we talk about when the blood's actually leaving the heart. Okay. And so what we see here is we see systole and diastole, so contraction of the heart and relaxation of the heart. And we can track different kinds of changes here. So we track pressure changes over time. We see that pressure in the artery, is here around 80 milligrams of mercury, increases during systole, and then slowly decreases during diastole. Where the left ventricle, though, has increased pressure during that systolic phase and then no pressure in that diastolic phase. So really here, the systole and diastole is tracking the pressure in the left ventricle. Okay. In terms of volume changes, it looks like this, right? So our ventricle is filling up, and then the ventricle contracts, pushing all that blood out, so the volume decreases. And then it's going to increase again as it fills during diastole. So we're seeing contraction is where you have ejection of the blood, and diastole is when you have filling of the ventricle. Okay. Looking a little more closely at diastole, now we have that isovolumetric relaxation, so rapid filling and atrial contraction, but the atria are smaller, softer, not causing the amount of pressure that you see when you get a ventricular contraction. Okay. Okay. So what do you think we call that little bump in blood pressure? The bump in blood pressure is when what happens? Number two, what causes the first and second heart sounds? Remember your lub dub? What's creating those? And the blank contraction creates enough pressure to open the valve and push out most of the blood. Okay, so now we're going to talk about the conduction system of the heart. Probably in anatomy you learned or you can recall about all the different parts of the heart and a little bit more about this systole and diastole. And this is controlled by this conduction system, okay? So first you have this something called the sinoatrial node, the SA node up here at the top of the right atrium. That SA node sends electrical signals to contractile cells in both the left and the right atria. This causes sodium channels to open and leads to contraction. So this is muscle controlled by electrical signals, right? So those principles we learned in the nervous system and in muscular contraction are going to apply here as well, okay? So once that sodium channel opens and contraction happens, that signal then travels from this SA node down to the AV node, so this atrioventricular node that rests between the right atrium and the right ventricle, okay? And there's a bit of a delay here. Then that signal travels to the bundles of His, the apex, and lastly the Purkinje fibers that you see travel down here, down the septum, between the ventricles, and out both sides. Only the yellow cells are the autorhythmic cells of the heart, okay? So those ones are autorhythmic. They beat on their own pace. The sinoatrial node typically sets the heart rate. The AV node and the Purkinje fibers are going to act as sort of backup systems. And of course, they carry the signal through the heart to create the contraction pattern we need to properly eject blood from the heart. Okay. So these autorhythmic cells, this SA node, the AV node, and the Purkinje fibers, make electrical signals and command the contractile cells. That sinoatrial node has about a 70 to 80 depolarizations per minute. Okay, so this is how it's signaling to contract. The AV node is about 40 to 60 depolarizations per minute. So if that SA node fails, this AV node will continue to be autorhythmic, continue to cause the heart to beat, but at a much slower heart rate. Okay, there's also a 0.1 second AV nodal delay that ensures the atria fully contract. So we need the atria to fill, the atria to contract, and then and give that a little bit of time so that the blood can move. Okay, so we said then the signal moves down the atrioventricular bundle, the bundle branch, and then finally the Purkinje fibers. And the Purkinje fibers have a depolarization rate of about 20 to 40 depolarizations per minute. So again, a bit slower than the AV node and the SA node. But these are the ones that signal the ventricular cells to contract. So we need a good pacemaker to coordinate events. If not, you get random excitation and contraction. This is called fibrillation. So typically that pacemaker is the SA node. But this is where if you're having failure of the SA node or other issues, you could have heart fibrillation. Okay, so here's what it looks like. Remember that, again, we're dealing with muscle. Okay, so we have to think about all of those same ions we did before in the nervous system and the muscular system. Okay, so these pacemaker potentials here are found below threshold. And then at threshold, we're going to get voltage-gated calcium channels opening, okay, causing depolarization, and potassium channels opening that are allowing a falling of that potential. Okay. So depolarization happens when they open the HCN channels. Then the cells can reach threshold. Our voltage-gated calcium channels open, and calcium coming into the cells equals more depolarization. Why? Because calcium is a positive ion, right? So any positive ion coming in is going to increase increase the membrane potential of the cell. The cells repolarize when the calcium channels close and potassium channels open. And just like in our neurons, potassium efflux causes repolarization, right? So loss of this positive ion takes us back to baseline. Then the potassium channels close and you can start again. Okay, let's think about a signal here. So your friend Rose tells you that her grandfather had a heart attack and had to have an artificial pacemaker surgically installed. It turns out her grandfather has a condition known as heart block. The cause of heart block is damage to the AV node. So let's think about these questions about heart block. What happens to the conduction of impulses to the ventricles during heart block? Would the rate of contraction of the ventricular muscles change during heart block? And if you were a cardiologist, how would you fix this problem? What could be done in order to restore some of the typical activity of the heart? Okay, let's look a little bit more at this action potential of a heart contractile cell. So here we've got sodium coming in, massive depolarization. Calcium can come back in very slowly, but then when you get a potassium leaving the cell, then you have a repolarization back to baseline. And you'll notice that the millivolts here is a little bit different than our neurons, right? So the typical neuron is going to be somewhere around -70. Typical heart contractile cell is going to be further down, so more like -85. You'll notice it looks really different as well, right? We're not getting that action potential peak like we see in neurons, and that's because of the flow of calcium as well. So we have this peak here from sodium influx, but we also see a slower decrease because calcium is also coming in slowly. Okay, so depolarization is that rapid sodium influx, so depolarization, sodium influx, plateau and action potential. And this plateau is when the sarcoplasmic reticulum releases calcium into the cytoplasm. At this point, those sodium channels close, and you have a delay in potassium efflux. For repolarization, potassium efflux, and calcium active transport back into the SR causes a decreased membrane potential back to baseline. Okay. So when we look at this, we can correlate the myocardial action potential and the contraction. This is what we see. So here's that action potential, and the contraction then is a little bit delayed. Okay, so the contraction follows the action potential, and we contract. We can measure this by tension developed. You'll note that the absolute refractory period of this is as this action potential is coming back down. Okay. And so essentially it's until just past the peak of the contraction. And the relative refractory period then is when this contraction is still coming down as well. Relaxation then is due to calcium pumped back into that SR and potassium efflux or repolarization. Okay. So this is a pretty long refractory period, and this is important because it allows full contraction of the contractile cells, ensuring adequate blood flow. It would be really problematic if you had a partial contraction of those ventricles and partial blood ejection. You need to fully eject all that blood from the heart and give enough pressure from that ventricle push that you can circulate it around your entire body. So we need that for adequate blood flow. And we don't see any tetanus or summation in those contractile cells. Okay. So finally, I want to talk a little bit about what an ECG or an EKG looks like. And we'll talk about this in lab too, as we do an EKG lab. So we'll get some more work done with this. But we can first look at the major segments of this EKG. So first is the P wave, and the P wave, this little bump here, correlates with or is caused by a depolarization of the atria. Okay. The QRS complex, this big thing that looks like an action potential, is when you get repolarization of the atria, so relaxation or diastole of the atria, and depolarization of the ventricles. So you have ventricular systole, atrial diastole during this QRS interval. Finally, you have a bit of a segment here as ST segment, and then a T wave. And the T wave is when you have diastole of the ventricles, so repolarization of the ventricles. And we can often see changes in the EKG, and change in the EKG can indicate certain pathologic or disordered conditions. Things like depression of this ST segment due to myocardial ischemia. Okay, and so this can produce lack of oxygen to the heart, which can cause this depression here when you have an ischemic condition. Okay. Other abnormalities we see in terms of heart rate and heart patterns is tachycardia, where you have a fast heart rate, bradycardia, where you have a slow heart rate, extrasystole, where you have a premature beat of the heart, and cardiac myopathies, things like heart muscle damage. What I want to point out here too is that some of these abnormalities might be symptomatic of a disease. Sometimes you can have some of these sorts of things as being environmental. Okay, so, or being at least, environmental at that time, and they might indicate further problems, right? I also want to point out that heart disease is the leading cause of death in the US, and so there's a lot in terms of cardiac function um, and cardiac work in clinic that would lead you to look at and read and interpret ECGs.